But now I'd like to invite Jukka Kallijärvi to give his talk. Jukka Kallijärvi is a principal investigator starting work, I think, next year at Folkhälsen, and he's interested in he works also in medical genetics, and he's previously had positions in Oulu as well as here in Helsinki, and he's also worked in Lund in Sweden as well. We're really grateful that he also could join us here to speak of his experiences in academia and in science. So please, Jukka, if you're ready, please take it away.
So good evening, everyone. It's nice to give a talk here in this event that I have never participated in anything similar previously. Maybe it has never been organized before, at least not in Helsinki. So I'm actually, David said that I'm going to start at the Folkhälsen Research Center, but I've been working there actually for quite a while already. First as about 9 years now. So I first started as a postdoc, doing my second postdoc. Then I progressed to a senior scientist, and then to something called co-PI, and then now working with the professor emerita who has retired already, and now I will start as a full group leader next year. So my main employer is a private research institute, although I have an affiliation with the medical faculty of the University of Helsinki as well. And this is our small research group, and our interest is in biomedical. It's in the field of biomedicine and medical genetics. We study complex 3 deficiencies, which are inherited metabolic diseases, also called mitochondrial diseases of mainly newborn children. And this is absolutely the best and most interesting project I have ever worked on. And since I realized that after having worked in this project with Professor Fellman for a couple of years, I realized that this is really an interesting, super interesting project, and exactly what I do. Then I have worked and we have worked together very hard to make it possible that I can become a PI and get my own funding and lead this research eventually. And I very much enjoy working with students, younger researchers, PhD students, on a daily basis, brainstorming, planning experiments, working in the lab, looking at results, analyzing, writing publications. I love almost everything about my work as a researcher. I even like grant writing. So very briefly about this, our research topic. It originates many decades ago, actually, when this Professor Vineta Fellman, a neonatologist and a pediatrician, started seeing these very, very sick babies in the children's hospital in Helsinki. And these babies that were luckily not born very often, maybe one or twice a year, were very sick. They had known condition that was clearly genetic, and there was not much that the doctors could do to help the babies. And then Vineta started investigating these patients and realized that this is a novel rare genetic condition or disease, and she published this in Lancet in 1998, describing this novel disease. And at that point, its name was not yet gracile syndrome, but in a couple of years she invented this acronym which describes the main symptoms or manifestations that the sick babies have. So they are very small, they have growth restriction, Then they have a kidney disease, which causes this amino aciduria, leakage of amino acids from the blood into urine. Then they have liver disease, and this cholestasis is a blockage in the flow of the bile fluids in the liver. So this is one sign of a liver disease. They have iron overload, which means that iron accumulates in the tissues of the patients, mainly in the liver, due to an unknown reason, and that may cause further problems. And then the patients have lactic acidosis, so their energy metabolism is defective in such a way that they produce too much lactate in the blood. It's this lactate is the same product that that your muscles produce when you exercise very hard, and you get muscle cramps eventually. And then the babies have a very short lifespan of sometimes only a few days. So our interest is, Vineta's interest has been for many decades to understand what causes this disease. And about 20 years ago, so it was obvious that this is a genetic disease, and it is inherited in such a way that it is probably a so-called monogenic disease. So it is caused by a mutation in one single gene. And then in about 2002, they found the mutation in a gene called BCS1L. And this gene encodes a protein that was then already known to localize in a cellular organelle called mitochondrion. And many of you, many of you who are not biologists or medical people will remember from your school books that mitochondria is the organelle in the cell that is perhaps the most important site for this energy metabolism. So the nutrients that we get from food, most of them are eventually burned or used for energy in mitochondria. And there are many other mutations. So this disease in the Finnish patients is caused by one certain mutation that has accumulated in Finland. But there are many other mutations that cause, mostly elsewhere outside Finland, that cause also these rare diseases of newborn infants, but they are mostly less severe than gracile syndrome. And then how we study this, what do we actually do? So this is a rare disease, and there are very few patients, and the patients don't live very long. So we cannot actually study the patients very much. There's very limited material from the patients. So we have to have experimental models to understand how this gene mutation causes this disease, and what are the disease mechanisms at the level of the mitochondria, the whole organism or the patient. And we use a mouse model that carries this same mutation as the patients. This was generated using this homologous recombination technology, with which you can put in mutations or so-called knockout genes in the mouse genome, and also in some other organisms. We use a little bit of fruit flies, and then we use cultured cells. And with these experimental models, we do so-called interventions that can also be called treatment trials sometimes. For example, we give different kind of diets, because this is a disease of energy metabolism. There's a problem in this mitochondrial complex 3. So it's been shown in some cases that modifying the diet can help patients with the mitochondrial disease. And then we have some other treatments. Then we analyze the outcomes of these interventions or treatments. We analyze the whole mice, measure their metabolism, for example. Then we collect tissue biopsies or samples. We analyze them, make tissue sections and analyze their histology. We do electron microscopy. We analyze proteins, enzyme activities. We do this so-called high throughput analysis where we sequence the whole transcriptome, for example, etc. So very many kinds of different techniques and expertise is required in this work. And then just quickly, I want to mention that I have interest in other metabolic diseases of children as well, and we have just started a new project where we study this LC Heart Deficiency, which is also a mitochondrial disease, but a very different, quite different one. So there's a problem in a gene that is required for the oxidation of fats, and this manifests as a metabolic crisis in the children, particularly if their blood glucose goes down or they get an infection, etc. And then there's long-term difficult symptoms or problems like heart disease and peripheral nerve damage. And there is a dietary treatment regime for these children with this LC Heart Deficiency, but it's quite tedious to maintain. And the disease mechanisms that cause the tissue damage in these patients are poorly known. So we hope that our studies will reveal more about this. So then the rest of my slides, I will go into my own personality and my family background. So the previous speakers have already talked a little bit about this family background. So I think that this is important. I want to talk about this because this is a very important contributor to why I have become what I am today as a person, and maybe also partially as a scientist, and also because this might be a bit surprising, and there's quite a lot of contents. Would be quite a lot of contents for some intersectional analysis. So I grew up in rural northern Finland in a working class family. I had a happy childhood, loving parents and very close extended families. My parents were not, they didn't and don't have any higher education, but they were fairly intellectual people. They read a lot. They were interested in the society and active politically. I was generally allowed to be the way I was, including some, as a kid, I mean, including some interests that were not typical of boys or that were considered maybe feminine. So I should mention here that I was born in 1973, so my childhood was in the 70s and 80s. My parents had a farm and various other businesses, so I and my brother had to work hard sometimes to help them. At the age of 10, I got interested in these STEM topics, mainly astronomy and chemistry, and later also computers. I had few relatives with higher or academic education, but the attitude towards education among them was generally very positive. And for example, my grandma had worked very hard during the 1950s and 60s to give as good as possible education to her 8 children, including my mother. My youngest aunt and an older cousin and several school teachers were role models and urged me to study further. However, these certain years, grades 7 to 9, were a very difficult time, and I was bullied at school, but I somehow survived. I was good at school, so there was something that I could that kept me afloat. So then from that on to secondary school and university studies. So I moved out from my home already at the age of 16 to attend a better secondary school, which was also in Rovaniemi in northern Finland, but further away from my home. I loved the school, but it was hard work. I took it very seriously. I had to have good grades from all subjects, and then together with the mental processing that was ongoing in my head related to my sexuality led to a period of depression at the age of 18 to 19 years. But then that resolved when I came out to my parents at the age of 19. And already at that time, I learned to know I immediately joined the local gay activities in Rovaniemi. If you can believe, there was a club or association there in northern Finland already. It had just been started in 1992, I think, and learned to know other gay people. I started my university studies with biochemistry being my major in Oulu University, and then I continued in Helsinki a couple of years later. At both universities, I found the atmosphere very liberal and inclusive, and I never felt any discrimination from fellow students or teaching personnel at these universities. And I was also somewhat active in the University of Helsinki Gay Student Association in the 1990s. Then my career in academia, I finished my PhD in 2006, and during my PhD and ever since, I've been quite out at work, although I have quite rarely brought up the topic, my private life with supervisors or colleagues, unless we have been somehow friends outside work, or sometimes someone has asked me about it, normally out of curiosity. I have never felt direct discrimination for being gay in academia. I have experienced quite a lot of other bad behavior and treatment and disdain and exclusion during my career as a researcher. But I think that those are more general problems of the scientific community, like bad leadership and bad social skills, the fierce competition and lack of penalty for bad behavior, and general meritocracy. And I think that being gay or a member of a minority group has given me positive perspective, and more ability to be considerate towards others, including my own students. That's all I